Hello, everyone. Thank you for tuning in to our fourth installment of our TDT talks. We really appreciate your time. Today we have David Barker from Rutgers University speaking with us. He's an assistant professor in the Department of Psychology, and so we're looking forward to the talk he's going to present. Just really quick housekeeping, we will have a Q&A session at the end of the talk, so there is a questions bar, so you can just type your questions in and we will answer them in the order they're received at the end of the talk. And if you are interested in having more in-depth discussion, we can host an additional follow-up coffee talk sometime next week for a more informal discussion. So just stay tuned for that. So without any further ado, I will hand things over to David and let him get started.
Thanks, Jessica. And thank everyone. I'd like to thank everyone for showing up today. I think this has been one of the huge benefits, if anything, of this pandemic. And one of the benefits for us has been having the time to work on some of these tools that I'm going to talk about today that might normally become kind of side projects in the lab. But when you're stuck at home, coding is something you can do pretty easily. So I was asked in giving this talk to break it down into two pieces, and I think it makes a lot of sense. So we have both a technical component, and I'm going to talk about this photometry modular analysis tool that we've been developing called pMAT. And then I'm also going to talk about a little bit of the science that's being, some of the work that I've been doing using this analysis pipeline. So I want to start at the beginning. I think many of you are probably here because you know something about fiber photometry, but for those that don't, fiber photometry is just a method for measuring time-varying fluorescence using an implanted fiber optic. And this started out with calcium sensors, but has expanded to an entire array of neurotransmitter sensors. And we're recording from volumes of tissue, so whole populations of cells. And we often combine this with behaviors, and it's great for freely moving animals. The sensors that we're able to record from, like I said, have expanded exponentially. We started out with GCamp, which measures calcium influx to cells, and that's had some different iterations, including red variants. But now we also have sensors for neurotransmitters like D-Light and GRABDA for measuring dopamine, or the iGABA sniffer for measuring GABA, the iGlu sniffer for glutamate, the iAtch sniffer for acetylcholine, or the iSero sniffer. And so these are just examples of all of those different sensors in one place, each of them time-locked to behavior. And I have to thank the very generous community of Twitter for providing me with a lot of these examples. And just like I believe in these open source tools I'm sharing with you today, we clearly have a very good community who likes to do that as well. One thing that I think would help is for me to just show my photometry system, or at least a schematic of kind of what it looks like. So this is probably also a very good time for me to say, I have no special interest in TDT except that they've earned my respect over the years by providing really great support. And you can also tell that from the hodgepodge system that I have. So we use Doric LEDs as well as photosensor and optics through them to kind of connect to the animal and perform these recordings. And then kind of in the middle of everything is our Tucker-Davis system, in my case, the RZ5 and Synapse software. And my lab is interested in all kinds of freely moving behavior like drug self-administration or pain, as I'm going to talk to you about today, is kind of a new direction for us. And so what we do is we create kind of a go-between between our behavioral system and the Synapse and Tucker-Davis system, where all the inputs and outputs from Med-Associates are collected as they would be normally. This is our behavioral system. But then what we do is we use this DIG 726 TTL card that has the same exact DB25 cable that the Synapse has, but they don't have quite the same connectivity. So we make a custom cable to let the two talk to each other. And in this way, we just mirror all of our behavioral events and put everything on one clock. So for those of you interested in doing this kind of setup, this is what mine looks like. For me, fiber photometry and starting to use this around 5 years ago, wow, time flies, is that it was the right tool for me at the time. So I like the tool because it's high throughput and you can have genetically defined targeting, but there are many tools that fall under this category. But for my needs, circuit-specific targeting was really important. We were trying to look at the function of a specific circuit in the brain. And then also compatibility with other techniques was really important for me. My background is as a single unit electrophysiologist, and many people have combined things like optogenetics or electrophysiology with fiber photometry. Lex Kravitz's lab has a great paper combining the electrophysiology, and Tina Kim from the Dijkstra lab had one of the first papers I can think of for optogenetics. And also very important for me was being able to record in deep brain. The lenses used for single cell recording didn't used to be able to reach as deep as I needed to record even in the mouse brain, at least not very easily and not with much clarity. Multi-site recordings are possible, and this is an advantage for many. And then the analysis is quite a bit simpler than the single cell recordings using mini scopes or things like that. One of the places where I think fiber photometry really shines is to be able to record at axon terminals. And I suppose it's possible you could do this with a mini scope, but it would be kind of a waste of the technology, and fiber photometry is really great for those kind of setups. Of course, inherent in some of the pros that I've talked about are also some corresponding cons, low spatial resolution. And one of the things that we found is recording at terminals is possible if they're dense, but not always guaranteed. So I think probably somewhere between 50 and 75% of the targets we've looked at, we're able to do this. And we've looked at a lot at this point, so I think that's a pretty good estimate. From my background, I think the time resolution is really poor. So if you're interested in things like auditory physiology, this is probably not the technique for you. And then one of the things that we know is a major con is that population activity, especially if the population we're recording from is heterogeneous, might only represent a subset of what's going on. For example, you might catch only increases and miss decreases unless the population is very homogeneous. And then where we come in today is that the analysis, even though it's simpler than the single cell, is still very challenging for many users. And the analysis approaches are not standardized. So this is where we came in with pMAT, which is just a GUI or graphical user interface developed around our analysis pipeline to try and make things easier and to bridge that gap. So the name pMAT comes from our development in MATLAB, and also pays a bit of tribute to the proteins for packaging neurotransmitters into vesicles like vMAT or vGAT, and also highlights the modularity of the tool. And this is just an overview you can see here, and we have a few different modules, each of which has a different function. So we have kind of a major control module for loading in data and for running everything that's selected if you want to run everything at once, or batch processing and kind of just taking control of everything that's going on. And then a number of other modules I'm going to talk to you about in detail. So some of the key features of this is that it's deployable in MATLAB, and that's something we wanted to retain so that we can have contributions from the rest of the community. People that do know the analysis can help expand this to future modules with other analysis approaches. But we've also deployed this as an independent executable file. So if you don't have access to MATLAB, this allows this to be an open source tool that anyone can use. It allows for processing of single files, and right now we're working on batch processing for other formats, for other maybe recording setups. But right now, you can do batch processing if you're running on the TDT system, which is great. If you have 20 or 30 behavioral files, you can run them all at the same time if they have a common setup. Then the system was designed thinking about some of the things that users need to do for both the analysis of big data sets, but also things you might need to do on a daily basis, like exporting and graphing data just to validate the quality of recordings at the end of a behavioral session, which is something that's very important for new users especially. And then kind of the main event here is being able to then examine these signals that you're collecting around relevant behaviors. So being able to produce whole session traces and see where the behaviors fall kind of over a very long-term time scale, or creating much smaller snippets that we call PeriEvent histograms or heat maps, where you're also able to look at the data that you've collected as it relates to behavioral events. And then, of course, as I showed you very early on in the talk, we have compatibility with a wide range of tools. So many different labs gave us examples of sensors, and the large majority of those we were able to run through pMAT and create some of the images that I showed you of all of these different neurotransmitter and cell activity sensors. So for some of the nuts and bolts, one of the things that was very important to us was bringing in cross-compatibility for individuals working with collaborators, or of course you might hand this off to students so you can load and save settings and also set prefixes if you are running different versions of one file, trying things in different ways. So there's some ability to change your settings here. And then also there are many times where we've worked with other labs that want to take their data out and they're using they're getting data from one of these systems and they want to export it very quickly to analyze in Spike 2 or some other system. So we've included compatibility for that. Probably most importantly is we've created this plotting trace module, which is meant to be a stepwise way to go through the control and signal channels that are common to most setups. So you can plot each of these independently, either the signal or control, and then you can also plot them in comparison to each other. And there's kind of a number of steps we go through in this analysis pipeline to get from these separated channels to a fit channel or the delta F over F, where we can start looking at behavioral events. For most labs, this starts out with a very high quality recording, but the signals that you're collecting tend to be too fast for most of the sensors we're looking at. So we often filter to remove high frequency noise. And then we want to do a few things to eliminate the photo bleaching that happens where the signal becomes a little bit, or the amplitude of the signal drops over time. And then the last thing we want to do is to be able to account for behavioral noise. So there are a couple of different approaches here, but I think one of the more common is to scale the control channel signal up to the level of the signal you've collected from your neurotransmitter sensor or otherwise, and most people do this using a regression. And the idea is to create kind of a predicted version of this signal channel. In this case, ours is 490 nanometers, so that's what it's labeled here. But the predicted channel is basically what should my signal channel look like in the absence of any of these neurotransmitter or cell activity specific events, right? What does background noise look like? And so a regression allows us to put these two on the same scale and create what the background noise should look like. And this is probably a little better example of that that comes all the way from the Fiber Photometry workshop I attended 5 years ago. It was probably the second one ever offered. And what this shows is that once we do this scaling, we're able to take any sort of artifacts that might happen in the signal channel. You can see most of these show up as deflections when an animal moves around and the fiber bends or things like that, and we can subtract them out to get a signal that only reveals the GCamp or eye sniffer or whatever thing you might be recording with. And in the end, we're left with a much smoother signal that doesn't have any of this kind of decay over time anymore, and we're left with just the final delta F over F set of events. And so we've described some of these things in detail in our preprint, but that's kind of the basic steps that people go through, and pMAT lets you see these piece by piece, so you can start understanding how they come together to create the final data that you might be seeing in other people's papers. And of course, the last and kind of most important thing for individuals interested in behavior is the ability to produce these PeriEvent histograms or heat maps. So the program kind of auto-populates with the different behavioral events. Oftentimes they have different tags if they're through the TDT system, or they can have whatever tag you want if you're importing from elsewhere. And this allows you to set a window of time before and after the event, as well as a baseline window that happens before this for normalizing the data using robust z-scores. And that allows you to generate these PeriEvent heat maps that show the different trials of the behavior all aligned to where you can take a look at what the activity of your sensor looks like around the time of that behavioral event. And you can start by just creating these basic PeriEvent histograms, but you can also export data for closer looks. For example, if you're interested in something that might be happening trial by trial, like how the signal changes over trials for learning, you can export the data that happens in these heat maps, export the average data for plotting somewhere else like Prism or Excel if you prefer. And then it takes you all the way to summary data. And probably the two most common approaches are to look at the area under the curve here. And you can set that for up to 6 different time points. You can also get the peak, which is something that others have used in different points throughout the literature. Here's just a little bit bigger of a view because I know that's a little small, but you can see we have these heat maps with trial by trial data, and then the kind of amount of heat that you have, the hotter the color, represents higher values for the signal being returned by the sensor, and lower values or cooler values represent lower values. And this all comes together as an average across trials where we can then see if an event is relevant. For example, in this case, we have a tone that comes on at 0 seconds and a shock at 5, and we can see there's not much of a response in this particular population to the tone, but there's quite a bit to the shock. So that's kind of a general overview, and we set this up starting at the most basic level, but one of the things that we're doing is we're setting up to import from other systems. So we have a generic CSV import that can work, but we've been working with a lot of other labs and very glad to have their support for getting data from Neurophotometrics, TeleFIFO, which is a new wireless system from Amusa, and the multi-site system, also called the FIP system, that was developed by the Diceroth lab in 2016. We're also working very diligently. In fact, I've got one meeting later this week to kind of finalize this on what you call a peak or a transient detection. Some people call it spike detection. That's not my preference as an electrophysiologist, but we've thought a lot about the problems associated with this and kind of talked with people that I respect that have been working in photometry for a long time and have come up with some ways of detecting spikes and giving the user some ability to decide things like thresholds and how to define the onset and offset. And so we think we're at a point where that's finally ready or nearly ready. We're also working on including detrending of the data that doesn't use a control channel, because some systems have that. For example, this wireless system from Amusa doesn't use it. And we're trying to integrate with other behavioral analysis tools. So you might have more complicated behaviors like licking, so we're working on a bout analysis for behaviors that need to be grouped together and a way to do that within the system. Compatibility with a lot of the tools other people are using, like Simba, which starts with deep lab cut for defining behaviors based on video tracking. And then we also know that many people have variable interval events. So you might have an animal hear a tone, and then sometimes they take 5 seconds, sometimes 10 seconds to go over and make a lever press. And so we've come up with a way where you can align these events independent of time so that you can kind of decipher the contribution of one event like a cue versus the time of the head entry. And so these are some of the things we're excited about pushing forward, and clearly the proof is in the pudding. You can see all of the bits of data here that we're using to develop those actively. So the last thing you might be thinking for at least this kind of technical portion is, where can I find all of this? And so you can look up the preprint, which will lead you to the GitHub, and also our lab website has all of these things, and most of them will lead you in a full circle back to either the GitHub or the preprint anyway. So I wrote them down a little bit, but I have to thank the members of my lab, Carissa, Chris, Svetlana, Jenny, and Karina. A couple of them have moved on. Carissa and Karina were great undergraduates who worked on pMAT and were incredible helps with that. And of course, my colleagues at the National Institute on Drug Abuse, Marisol Morales, my former PI, David Estrin, who helped with the pMAT code, along with a lot of the data collection for the snippet of data I presented today. And there are many, many more people at NIDA that also helped with other aspects of this project I didn't have time to present today. And then Dr. Chris Town, who really helped us with the sciatic nerve approaches that we started using, Alyssa Margolis and Kate Pena, who both been working on other aspects of pain work that we've started on over the last couple of years as I've started my own lab, and then my funding sources. With that, I would be very happy to answer any questions that you might have.
Thank you very much. That was an excellent talk. Yeah, so once again, feel free to type your questions into the question bar. We already have one from Barish. She said, very nice tool, pMAT. Regarding compatibility with video files, do you have code to align the two data sets, FP and video, as well as upsample the video data set? Sampling frequency is likely to be much lower.
Yeah, so one of the things that's included, and I guess the answer is that this depends on the system. So right now we pull in all of the video timestamps if they're coming in from a TDT web camera or other things. So you could align things, and so if you knew specific video timestamps, you can bring those in. Another thing that we added is the ability to append timestamps. So this came directly out of a collaboration back with NIDA, with Dr. Flavia Barbano, who had some video scoring in. So she gave me timestamps and we put them in as a CSV file, and you could bring them in from your own kind of manual video scoring. So it all depends on the approach, but the hope moving forward is that things like the Simba analysis pipeline or other things, we can bring in relevant video timestamps. I don't have any plans right now to support the analysis of video within the pMAT suite because I think others are doing it just better than we can right now.
All right, next question we have is from David. Thanks, David, great tool. Does this tool also process other time series data like spike trains?
That's a very good question, and one that I honestly hadn't thought about at this point in time. I think that's a great idea for a future direction, especially because one of the reasons that I personally use the TDT system is because I'm also an electrophysiologist, and also because a fun fact is that the original system, once they got away from really expensive machinery, was developed on a TDT pipeline. So I think that's a great idea. It is not something that we have compatibility for, but it would be something that would be very easy once you've exported something like your online sorting or cluster cuts. You could bring them in as timestamps, and you could see how the two signals relate if you happen to have the two. It would be fairly easy for us to maybe create compatibility for the PeriEvent histograms also. That would be a great either secondary tool or future direction. So thanks.
All right, we have a question from Cesar. He said, hi, nice talk. I'm using a similar system, but I use Gcamp and mCherry. So I started using interleaving acquisition of the signal, on, off, on. Would the system be usable in that case? Thanks.
Absolutely. So in fact, we've been working with Cody Siciliano, who's got the kind of multi-site system that uses the same interleaving, and we have some data that's in that format. Right now, if they were entered in as two separate channels in the CSV import that can be used with really any format, you would put the mCherry in one column and the Gcamp in another, and it would treat them just like the signal and control channel here. I know a lot of people are using static fluorophores like mCherry as controls, and I think the system would handle it just fine in that way. So maybe you have to do some things to get the formatting in for now, but as I've said to anyone, please reach out if you have kind of a specific way that you're recording your data, and we'll see what we can do to increase the compatibility of importing other styles of data. That's a really important goal I have for making this tool as widely compatible as possible.
All right, from Michael, he said, very nice talk. For the fiber photometry recordings you showed in your pain study, were the comparisons between subject or within subject? I'm trying to understand limitations, difficulties with comparing FP traces across different animals as opposed to within subject comparisons.
Yeah, so when we're making comparisons. What we do most often to decide whether an event is relevant is that we will look at a window that is a baseline window within subject compared to an event window within that same subject. But we also do comparisons across animals. And this is actually one of the reasons that we've moved over time from looking in the lateral habenula in 2017 to our recent paper looking at different kind of neurotransmitter defined cell types in the VTA that was just released a week or two ago, to using these robust Z-scores to make comparisons across animals or groups a little bit more relevant. There are some definite caveats there, but I think that if you're normalizing and that you're checking the quality of your signal to where you're not comparing a group that has very terrible signal with a group that has very good signal, which is part of why we included this stepwise plot tracing, then you can do either of those, and both are equally relevant and equally okay.
All right, John said, thanks, David. What are your thoughts on trying to generate a closed loop system that would use the on-fly analysis to trigger additional stimulus or opto-channel?
Yeah, funny story. I wrote a grant last week that I wasn't eligible for on exactly this idea. I love it. I think it's a great idea. And then I found out I needed tenure to submit it. So we all waste our time every now and again, right? But I think this is an incredible idea. And I actually, something that gives me hope is this Amusa wireless system. I think something like that would be perfect. It comes from a Japanese company that they work with that already has a wireless opto and a wireless ephys. I think there's a lot of work to be done, but I also think that would be a really great tool to have to be able to do this sort of sorting. One of the biggest problems for doing that with fiber photometry is that a lot of these fancy calculations I've been showing today would need to be done online in order to at least keep track of some of the local bleaching and to try and make a decision about spike detection, which, as I said, is one of the most kind of hotly contested things for photometry, because, right, we're not really measuring spikes, we're measuring increases in calcium that are a proxy for neural activity and might, in the case of photometry, be 10 spikes or 100 spikes. So I think it's an awesome idea. I encourage anyone that's interested to do it, because it takes a village, right? But it's a dream and not anything I know about as a reality right now.
All right, TS said, is the Noxious laser stimulation in the paw achieved just by the floor lighting up, i.e. the light shines through the paw and stimulates the neurons?
That's absolutely right. So this channel rhodopsin transfects neurons in the dorsal root ganglion and gets trafficked out to the free nerve endings in the paw. And then you activate those by shining blue light that goes through the paw. So you need a little bit higher power in some cases, but even with a floor full of LEDs, we were able to get pretty nice results, and they're very consistent with a response that is painful. Like I said, that flinch you get if you were to touch a hot stove is what we see in a mouse.
Oh, another question. John was asking, will this photometry technology replace the multi-channel recording?
I feel like I don't want to touch that one with a 10-foot pole, but my best estimate is that no, because the example that I gave in the middle of the talk, I think, is a great one. I've got a really wonderful colleague in the lab next door to me that does auditory physiology, and there you need really rapid responses, and really the only appropriate tool for that is either single or multi-unit electrophysiology. The photometry is simply not fast enough. Voltage sensors are a thing, and they work, but not as well as some of these other sensors that I've talked about. So maybe we'll hit a point where the voltage sensors will be a replacement, but at least for now, I think the single and multi-unit recordings are safe.
All right, Priscilla asked for the Paulite stim, how long is your stim? Is a 4 seconds withdrawal latency short? I'm not a pain expert. It seems long to me, not sure if you need a long stim to get enough chondroitinase activity, or because it's a polysynaptic circuit.
Yeah, I think that it takes a little bit more power and more time to activate the system, and there's a pretty big range too. And we think this relates back to number one, us doing this for the first time and maybe not being so good at getting a needle into the sciatic nerve of a mouse, which is big but not that big. So we might have some more variability, but we see polywithdrawal times that are nearly instant, fractions of a second, all the way up to a few seconds. So I think that's a big part of it. And I also think, yeah, getting the correct light power to the paw is a part of it.
All right. Well, I think we have wrapped up with questions. If anyone has any more that they would like to submit, now is your time. I'm just going to do a quick shameless plug because one of the questions was about doing closed loop, a full closed loop system with fiber photometry. And on the TDT side, we do have a new gizmo we've released called pMAT, which allows you to integrate Python coding directly into the TDT system and Synapse software, so you can directly control not only your behavioral side, but pretty much what, the main things you can do on Python, you can now do it directly through the TDT system. So our hope there is to just decrease latency times and have everything in one place. So that's definitely something to consider. Maybe will help get a little bit closer to, to being able to do the real-time coding for a closed loop system with fiber photometry. So that's my shameless plug. And it does not look like we have any other questions. So I think with that, one more. Oh, yeah, so that's for the, so TDC system is open for Python scripts from John. Yes, there is a few modifications, but actually, John, if you want, I can chat with you offline and let you know a little bit more about the new pMAT. So, all right. So I think that about wraps up everything for today. So David, thank you very much for taking the time to present your work. It was really fascinating, a lot of fun to learn more about. And with that, I want to thank everyone for attending. And David, do you have anything else that you wanted to say to wrap up?
Yeah, no, thank you for having me, and thank you everyone for taking the time out of your day. I really appreciate it. And I guess I'll say, if you want to, you can, you've got the website through the talk and other things. You can reach me if you have any questions beyond this, and I'm happy to help.
All right, thank you very much. Have a good rest of your day.